Hello, welcome to 13 Universal. So this is A-level CIE Biology Advanced Practical Skills P3 2024 exam checker May June 31. So if you are doing May June 31 2024, this is an exam checker for you. It helps to predict likely questions coming out in May June 31. Okay, stay with me. This is schedule. Come on, get your writing materials. Let's dive into the questions. All right, so this is one of the questions that, of course, are going to be solving questions that are related to what is going to be coming out in May, June 31 here in this region. The exam date is 9th of May. Okay, so watch this video and get ready for the exam. Okay, look at this. The first question: onion cells in sodium chloride solution may show plasmolysis. So what is plasmolysis? Detachment of cell membrane from cell wall, where the cell surface membrane pulls away from the cell wall. So the cell wall and the cell surface membrane are fighting. These guys used to be friends and they got married and now they are separating. Oh my God, what caused this problem? Let's go. Fig 1.1 shows onion cells in sodium chloride solutions. Okay, shows in sodium chloride solutions may show, says fig 1.1 shows onion cells in sodium chloride solutions may show plasmolysis. Cell A is showing no plasmolysis. Okay, how they show plasmolysis? That's what it means. That's a typo. Okay, cell B, C, and D are allowing plasmolysis. They are all showing plasmolysis. Look at it. A shows no plasmolysis. Okay, the cell is intact. Cell membrane and cell wall are still together. They are married. All right, cell B, slight plasmolysis. Now they are beginning to separate. They are having issues in their marriage and they are beginning to detach from each other. Seeing counselor is not working. So now this is detaching. You know what? Why is it detaching? So the first question I'm asking you is, why does cell membrane detach from cell wall? Of course, you can see cell C, more plasmolysis. The cell membrane detaching more, and then you see fully detached. Okay, complete plasmolysis. So why does cell membrane detach from cell wall? Okay, when does this happen? Okay, this happens when cell loses water, when a cell is placed in a solution that has lower water potential than the cell content. Water leaves the cell, and the more water leaves the cell, the more the cell plasmolyzes. So I have a theory for you, hypothesis. Okay, that theory says that water potential, remember that water potential is indirectly proportional to solute potential. So I use the word concentration. All right, concentration. Let me use the word concentration. So the more concentrated concentration, I put the word conc here, is indirectly, or directly proportional to plasmolysis. All right, plasmolysis. So don't forget this. You're going to need this as we navigate through these questions. Okay, so yes, this is plasmolysis, yeah, that I'm writing. Plasmolysis, lysis, process where cell membrane detaches from cell wall. So this is the relationship, guys. Okay, so the more concentrated a solution is, remember that, guys, we are putting the cell is being put, now this is a beaker, right? Beaker. So put the cell here, okay, from a tissue, see the cell put here, okay? All right. So this is the solution. Okay, we submerge this cell inside the solution. Okay, so don't forget that we're submerging this cell inside this solution. Okay, so we have the concentration of the solution. All right, so as the concentration of the solution increases, that means it's going to have lower water potential. It's going to have more negative water potential. The higher the concentration of solution, the more negative the water potential is, and then the more plasmolysis occurs. So if you look at this cell, if you look at the cell A, this cell S seems to be placed in isotonic solution. Nothing's happening here. Okay, see, but cell B means that the solution is becoming hypertonic. That means the concentration of solute in the solution, right, is becoming high. That means the water potential in the solution is becoming, less compared to water potential inside the cell, right? So as the concentration of solute increases, all right, the plasmolysis, the level of plasmolysis also increases. That means this will have less, okay, solute concentration compared to this. This has more solute concentration. C has more solute concentration than B. That is why we have more plasmolysis. And D has a higher, what is it called, solute concentration than C and B. So if I have, for example, now I'm just introducing this because it's very important, okay, introducing part of this video. So if I have solutions like one is 5% concentrated, I'm just assuming, all right, this is 4% concentration, that's solute concentration, and I have like two, I have like three, okay, let's say three here, 3%, I have two. My question is, all right, which of them, and of course I have one, I have zero. Okay, which one will appear like cell D? Probably between 5 and 4. Okay, cell D. Which one appears like cell C? Cell C. Cell C probably 3. Okay, let's say 3 and 2. Cell C. Okay, then cell B most likely, all right, number 1. Okay, that's 1%, and this is A. Okay, or less than that. Okay, so what I'm trying to say is that the higher the concentration of solute, the more plasmolyzed. Right, let's go to the next question, next part of this. Okay, now look at the next question, because we're following the trend here, the instruction. It's a different concentration of sodium chloride solutions have different water potential. You can see that. Yeah, so that sentence is very important. So different concentration of chloride sodium chloride solution have different water potential. That means the higher the solute, this concentration of sodium chloride in a solution, all right, the less the water potential. When onion cells are put into sodium chloride solutions with a lower water potential than the cell, yeah, water leaves vacuoles and cells show plasmolysis. Yes, let's go. You are provided with materials shown in table 1.1. These are the materials, guys. See, labeled. These are the labeled material. This S1, S2, S3. So if you're in the lab working, before you start working, guys, compare what you have on the bench with what you have here. Okay, because this is a practical exam. All right, so check, do I have anything labeled S1, do I have anything labeled S2, S3, S1? Of course, this is telling you the content that this has 0.5 mole per liter of sodium chloride solution and onion tissue, so all of them, all right, but this is known. We don't know S2, S3, S4, but they all have sodium and chloride concentration, but we know the concentration of S1. Now, the hazard, none of them is hazard, so don't be afraid, okay? None of them will cause injury or harm. These are the volumes. So let's go, guys. Yeah, if any solution comes into contact with your skin, wash off immediately under cold water. It is recommended that you wear suitable eye protection because of the onions. Onions can make your eyes produce tears, okay? So you can wear eye protection. S1 is now disclosed. S1 is 0.5 mole per liter cube sodium chloride solution. So this is known. Now S2, S3, and S4 are 1.00 mole per liter cube, 0.25 mole per liter cube, and 0.00 mole per liter cube, but not necessarily in that order. So we don't know the order, but we know that this, all right, represent this. We don't know the order. You will need to observe a sample of onion cells from S1, S2. So we need to observe this S2, S4 using microscope. Decide how many cells you will observe to record the state of plasmolysis. Record the number. So this is what we're going to number. Now, guys, let me say this. In every scientific experiment, there are 3 variables you must know. So as you walk through these, this procedure, in your mind, you should be thinking which one is independent variables, which one is my dependent variable, and which one is my key variables. The two important ones are independent and dependent. Okay, independent is what is being changed. Okay, you can see that what is being changed here is concentration. Either it's 0.5 or 1 or 0.25 or even 0. But what are we going to measure? Because it's a recorded number of cells. So the number of cells that show different states of plasmolysis, okay, is what we're going to record. Use your result to identify the concentration. Remember that the higher the concentration, the more plasmolysis will occur. All right. Use the result to identify concentration of sodium chloride solution. S2, S3, S1. We're never told the exact. Okay. So let's jump in. Carry out. Remember, in exam, stay with the procedure. Don't look around checking what somebody's doing. Carry out step 1 to step 7 to prepare a slide of onion tissue. Label one dry. So the first one you do is label one dry and clean microscope slide 1. Slide S1. Put slide on a paper towel. Put a few drops of sodium chloride solution S1 onto the microscope slide. Remove a piece of this onion tissue from S1. Peel off the inner epidermis as shown in Fig 1.2. You can say the inner epidermis may start to separate or have become separated from rest of the tissues. Okay, so look at it. You just remove the inner part. You can see that. So in exam, you have to follow the procedure. So just follow the procedure as being shown there. So it's being shown to you, so you can follow it. All right. So follow the procedure. Follow the procedure. Okay. So now, so what's the next thing to do? The next thing to do is watch this. All right. So you follow the procedure like it's been put out there for you. So you can see there. So you put, this is the microscope slide. All right. This is the paper towel. Put it on, and then piece of epidermis. Remove this. Okay, this instrument here. Okay, to remove the inner epidermis. Put it here. Okay, and then spread it. So the chloride solution. Right. So by looking at it, you will know what it's asking you for. Okay, so these diagrams guide you. They are stimulus materials on what you are supposed to do. So these are the other instructions. Cut one piece of the epidermis so that it will fit under a cover slip. So this is a cover slip. Okay, so yeah, so cut the one that, this is the one that will fit in. Okay, yeah, that will fit. This is slide, so this is your cover slip should be able to cover this, right? Into put the remaining epidermis into S1 where you brought it out. 5, put the piece of epidermis on the leveled microscope slide as shown in Fig 1.3. If the piece of epidermis is folded, you may need to add more drops of S1 so that it floats and can be unfolded. Now put a cover slip over the piece of epidermis or microscope. Now when you are putting it, make sure you put it at an angle, and then once you put it, make sure you push the cover slip in order to dispel trapped air. It's not going to have trapped air. They form artifact. Okay, use a paper towel to remove excess solution that is outside the cover slip. Observe the result of the epidermis using the microscope. So you put it under the microscope, all right, and you may need to reduce the amount of light, yes, using the diaphragm of the microscope under the microscope where the light is. So regulate it, reduce the light entering the microscope to observe the cells clearly. So these are the instructions. Now the questions, let's go. You need to decide which objective lens you will use and decide the number of cells you will need to observe to give you confidence in your results. Okay, I says state the objective lens you will use and the number of cells you will observe. So this is testing your ability to make decision. Explain how this gives you confidence in your result. All right, of course you need to, for you to have confidence in your result must be reliable. It must be show a level of accuracy. Okay, so you will need to see the cells that are plasmolized, right, clearly. So use a higher magnification, right, of objective lens. Use times 40. Okay, the reason is that so that you can see the plasmolized cells clearly. You need to see it clearly to be able to make judgment. Okay. Now, if you say use times 10, you may not see it clearly, but it may give you more number of cells. But times 40 will give you a clear view of plasmolized cells. And of course, you don't want to see too many cells, right? Maybe around 10 or 11, and the ones that enough for you to observe the change. Look at objective lens times 40 number of cells. Okay, maybe 10 or thereabout or less than that. Explanation to see the plasmolized cells clearly. Let's look at masking. Objective lens times 40 plus number of cells. Okay, cells is 5 or above number of cells. Okay, don't need too much. All right, but minimum 5. Idea of cells being large enough. Yes, that's why we're using times 40 to see plasmolysis clearly with times 40. Or idea of having a large number of cells. There are enough to count cells in different stages. Yes, we have more. You can see cells in different stages of plasmolysis. Okay, 2 marks. Using the objective lens and the number of cells you need in A1, count the number of cells for S1. So S1 we've been told 0.5 mole per dm cube that are in each state of plasmolysis. The ones A, B, C, and D are shown in figure 1.1. Record your results in AI. Repeat step 1 to step 8 for each of S2, S3, and of course S4. Record your results in AII. Very important. Okay, now record your results in an appropriate. So they want us to record our results. So I want you to pause the video and draw a table. If you are in the lab, how will your table be? Okay, yeah, don't forget guys that we use closed tables. Okay, your tables must be closed. All right, like this. Okay, it must be enclosed, right? Yeah, and then you must have heading. Okay, if of course what we are changing here is usually the first column, which is independent variable. Independent variable here is the different solutions. Okay, different solutions, though we do not know the concentration, only one that we know, so we use solutions. Okay, yeah, we don't know the exact concentration, we can call it solutions, all right, or test tubes. So this is it, solutions, no units, and of course number. If you check this, we counted number of plasmolized cells, so you see, but that a number of plasma cells in each stage, right? This is number of plasmolized cells. So number of plasmolized cells, okay, because that's what we're asked to count, number of plasmolized cells. So I may not write it fully, plasmolized, just this, but in exam write it fully, cells, yeah, plasmolized cells. So number of plasmolized cells here. So remember, in each stage I have about 4 stages, so I'm going to have column for each one, right? So you have to plan this table, because before you start imputing your values. Okay, so this is the one in stage A. How many cells are in stage A? How many cells are in stage B? Okay, how many cells are in stage C? And how many stage in stage D? Very important. And then of course we come to solutions. We have about 4 solutions to talk about. We have solution 1, okay, S1. So we have solution 2. So we put number of cells present in each of them. If not present, put 0, because this is S2. Yeah. Now solution 3. So we are predicting your exam. So this is most likely what you could be asked to do, right? Yeah, S4, can see. So you now put, once you view under the microscope, check the cells in stage A, no plasmolysis. Okay, yeah, plasmolysis starting, more plasmolysis, complete plasmolysis. Right, their numbers. Okay, okay. Let's see what the mass scheme says you must have. Answer. So you can see it said heading for solution or sample. So heading here, 1 mark. Okay, heading for dependent variable. So this is independent. Dependent variable, state of plasmolysis. Yeah, number of plasmolized cells, state of plasmolysis. Okay. Collect results for each solution. For each solution, I have result. Put it there. Correct trend. Okay. Record a number for each. Correct trend means the higher the concentrated, all right, solution is, the more plasmolized cells we're going to see. Okay. Record a number for each solution, each state of plasmolized, at least 16 number. 16 numbers in total. That's what we're looking at. Okay. So you record it for each of them based on what you have. All right. So we're done with this. Let's move to the next one. 5 marks, guys. 5 marks. This is your bench work. This represents all your bench work. 5 marks. Very important. Okay. So your bench work carry. So each mark has a point. This is this. The first point here is this. 1 mark, 1 mark, 1 mark. All right. That's how it's been marked. All right. Solution S1 is 0.05 mole per dm cube. S1 has been told. S2, S3, and S4 are these. Okay, 1 mole per dm cube, 0.25, 0.0. So any solution that has 0.0 mole has no sodium chloride, of course, present. That means there should not be plasmolized cell at all. The cell should be turgid. Okay, but not necessarily in that order. Use your result in AI. Identify the concentration of sodium chloride solution in S2, S3, and S4. Guys, whatever you have in your table before will be used to answer this question. So that means that whatever is S2, if S2 has more number of plasmolized cells, it will have the highest concentration. The one that has more number of plasmolized cells is one that has the highest concentration. The one that has no plasmolized cells, or the least number, should have the should have the lowest concentration. Do you get? All right. So if I have S2 as more number of plasmolized cells, that will be 1. Okay. S3 less number, 3. That will be 0.25. Then no plasmolysis. Okay. A few number, that's 0.00. So the higher this concentration, of course, the more number of plasmolized cells based on your table. All right, let's go. So you see it, correct result from candidate table. So from your table, you determine this. Your table determine this. All right. Yeah. So look at the next one. 1 mark. Observe onion cells on the microscope slide you labeled S4. The S4. Select your adjacent touching cells. Select 4 adjacent touching cells with at least 2 cells showing plasmolysis. 4 adjacent. Make a large drawing of these 4 cells. Use one ruled line and label to identify the cell surface membrane of one cell. Okay, so based on what you have, because you have to put the slider, of course, that has S4, and then view 4 touching cells at a higher magnification times 40. Guys, what you expected to have, right? They said they are touching cells, okay, with at least 2 cells showing plasmolysis. So the cells are touching. So you should draw something like this. Okay. Yeah, very large. Okay, this is one of the cells. Okay. Remember, each cell will have cell wall. Cell wall is double, represent using double line. Okay. The line shouldn't touch. Okay. Pardon me for this. I have to redraw it. Okay, because I have to show you how it's been drawn. Now you have to draw the cells that you see. Don't draw from your mind, because your examiner would have viewed this. Okay, draw the cell you see on the slide. Let's assume this is what we're seeing, but I'm showing you how it should be represented. 4 touching cell. Okay, cells. So this is another cell here. If they're going to be touching each other, they must assume a position here like this. Okay, each cell should have a cell wall. So each cell should have a cell wall. The cells don't have to have the same size, but they are touching. See, each cell should have a cell wall. Yes, let me put this here. So this is another cell. Each cell should have a cell wall. This is third cell. Okay, remember each cell should have a cell wall. Yeah. So because there are 4 touching cells, right? Here is another cell here, something like this. Based on what I see from the microscope, I'm drawing 4 cells, right? Each one has a cell wall. Remember that another instruction there is that at least 2 cells show plasmolysis. Okay, if I like, I can decide to, all right, maybe show a little bit of plasmolysis here, based on what I see. Okay, yeah, let's assume this is plasmolysis. Let's assume I have this level of plasmolysis here. Okay, let's assume I have a plasmolysis like this level of plasmolysis. Okay, guys, remember this is based on what you see from the your specimen. Okay, let's assume one is not plasmolized. Okay, so I represented it. Okay, remember each one has cell wall. At the boundary where they're touching, you have 3 lines. Cell wall is represented by 2 lines. Where they touch, 3 lines. Okay, yeah, so you can see lines are continuous. Remember, the lines you use must be continuous to get 1 mark. It must, now this is 4 marks under 4 points. The lines must be continuous, thin and sharp. Okay, minimum. So continuous line, draw only 4 whole cells, that's under mark, and each cell touching at least one of the other cells, at least 2 cells showing some degree of plasmolysis. Okay, all right, at least each cell touching at least one. So you can draw it like this. You can draw them in a straight pattern. Okay, cell walls as 2 lines. So cell walls are 2 lines, 3 lines where 2 each cells touch. So see, this is where cells are touching 1, 2, 3. Okay, right. So you could draw this like this. Okay, you could as well draw it, like this pattern. Okay, somehow like this, but you have to add a cell wall and draw it large. Okay, this is not wrong. You can draw it like this. Okay, but make sure that at least 1 cell touching 2 other cells. So put a cell wall and all of that. Do you get? Yes. So that is it, guys. So let's look at this format. A student investigated the effect of different concentrations of sucrose solution on pieces of potato tissue. Each piece of potato tissue was cut to exactly 50 millimeter. So you see, this is one of the constant variable, right? The length must be same and had a cross-sectional area. The cross-sectional area must be the same. Okay, and each piece of potato tissue was put into different concentration of sucrose solution. All right, after 1 hour, the length, so 1 hour is also key variable. They must all stay in the solution of different concentration of sucrose for 1 hour. The length is measured after that was measured. Now this is somebody's the result of the student's investigation are shown. This is somebody's. Okay, now this is sucrose concentration, which is independent variable, is causing change in length. All right, so at 0, this is the change in length. It has the highest 0.2. so as concentration is increasing, what's happening to change in length is decreasing. Okay, all right, why? Look at this question. Describe the pattern of water movement shown in table 1.2. So pause the video and try it. What is the pattern of water movement? Now, did not say describe the data. The pattern of water movement is deeper. So you can see, right, from 0, all right, to 4. Now this is 4. 4 means that the amount of water moving, because you see, if the length is increasing, that means water is moving into the potato tissue. So that's why the cells become turgid and then the length increases. But if the length is decreasing, because you can see that change in length is negative, that means water is leaving the tissue. All right, from 0.6 to 1, water leaves the tissue into solution. Okay, so water enters solution from the tissue. That is why the size of the length decreases. But from above, see, can you see concentration below 0.4? 0.4 is the mark. Concentration below 0.4. All right, water enters cell. Okay, concentrations above 0.4, water leaves the cell. So at 0.4, the amount of water entering the cell is the same as water leaving the cell. All right, let's check. So you see below 0.4 mole per day cube, that is from here and above, water moved into potato tissue. That's where it goes. Okay, now above, water moved out. Okay, no net movement of water at this. All right, so 2 marks. Now, a student investigated the effect of different concentrations of sucrose solution on pieces of potato tissue. Each piece of potato tissue was cut to exactly 50. I've read this before. The second question we're looking at. The result, yeah, that's it. Let's go to the second question. Yeah, using your knowledge of water potential, explain the result at 0.2. So, why is 0.2, right? Why is there increase? Pause the video and try your knowledge of water potential. So why does the change in length, all right, why does it increase? Okay, this is because remember that the water potential of the solution is higher than that of the cell content, right? So if the cell content is this, or the tissue, whatever you call it, okay, the water potential of the solution is higher, so water moves into the cell, okay, by osmosis. So water is moving from solution to the cell by osmosis because the water potential of the solution is greater than that of the cell content. All right, so it has a you call it less negative water potential. The solution has less negative water potential than the cell content. All right, so you see that the sucrose solution had a higher water potential. This sucrose, okay, yeah, the solution had so than the potato cell. So water moved into the potato, okay, by osmosis. That's what explains that. 2 marks. Okay, the student decided to compare the effect of different concentrations of sucrose solution using different types of potato. You can see that. Yes, there's a complete table 1.3 suggests 3 variables will need to be standardized. So what are the 3 variables that we need to standardize? Keep constant. All right, like the length of potato tissue should be the same. We've seen this, and how do we do that? You measure using ruler. Okay, you can also keep the what is surface area should be the same using the same ruler. Okay, potato tissue should be the same species, age, the same time, same temperature. Okay, look at it. So remember, this is likely going to come out. Potato, same age, because if any of these changes to affect the result, they must be kept constant. Okay, same potato from same plant, surface area, use ruler or caliper or cork borer, length using measuring equipment, ruler or caliper, volume, yeah, volume of each concentration should be the same. And these are the things you use: measuring cylinder or burette, pipette. Time should be the same. Time left in solution should be the same. Timer, temperature, use thermos should be the same, because temperature can actually affect the rate of diffusion. Osmosis is diffusion of water molecules. Okay. All right, 3 marks, guys. Yeah, let's quickly look at the next question. Amylase is an enzyme which hydrolyzes starch into reducing sugar. Okay, the second part of the question, wet practical, as shown in Fig 1.1. You can see starch is hydrolyzed, all right, by amylase to produce reducing sugar. Now, they said the progress of this reaction can be followed by measuring the concentration of starch remaining using iodine solution. You will need to prepare a serial dilution of 1% starch solution. Estimate the concentration of starch remaining after hydrolysis with amylase at 2 different pH values. You are provided with the materials. Now guys, before you continue in exam, as you are reading, you find that what is my independent variable? What are we changing here? What are we mentioning? Of course, what you are changing here is concentration, because you are diluting. Diluting is a technique to vary concentration, and whatever you are varying is independent variable, right? So whatever you are measuring, what are we going to measure? We're going to measure concentration of starch remaining. Okay, so remember, starch is a substrate, so the rate of reaction, all right, is must be inversely proportional to the amount of substrate or starch remaining. So that means that if the reaction is high, the amount of starch remaining will be low. Most of the substrate will be used. If the reaction is low, we have lots of starch, okay, available. Okay, therefore the color of iodine on starch. So let's look at this. You are provided with materials shown. These are the materials. Like I told you, if before you start your work, check whether these are provided for you on the bench. So S is 1% starch. This iodine, water. This is water. No hazard. Okay, this is the volume. X, a sample of 1% starch solution which has been incubated with amylase, harmful irritant. The amylase may be harmful, right? It's an irritant. So be careful with it. Wear gloves. Okay, a sample of 1% starch solution which has been incubated with amylase. Right? Be careful with this. And of course, if any solution comes into contact with your skin, wash off immediately under cold water. It is recommended that you wear suitable eye protection. Guys, protect yourself. If you don't have these protective tools, on the bench, raise your hand and ask the scientist, the lab scientist, to provide it for you, or the supervisor of the exam, because you need to protect yourself. Okay, these are the hazards, they impose risk factors, cause injuries and harms, and you have to take precaution as scientists. All right, now let's go to the next question. Next part, you need to carry out a serial dilution of the 1% starch solution S, which reduces the concentration of the starch solution by a factor of 10 between each successive dilution. So you see, so serial dilution by 10, that's tenfold. Fig 1.2 shows you the first two beakers you will use to make your serial dilution. Okay, so these are instructions. Complete Fig 1.2 by drawing as many extra beakers as you need for serial dilution. For each beaker, state under the beaker the volume and concentration. State under the volume concentration of starch solution available for use in the investigation. Use one arrow with a label above the beaker to show the volume and concentration of starch solution added to prepare the concentration. Use another arrow with a label above the beaker to show the volume of W, that's distilled water, added to prepare the concentration. Okay, all right, make sure you understand and follow it. So this is it, guys. Okay, this is we're suspecting that they will ask you to dilute something. Okay, either serial or simple. Now we're dealing with simple. This is serial dilution. Next video will deal with simple dilution on this. Okay, remember we're trying to predict the exam, right? So you have to add other beakers. You draw this in exam. It's about 3 marks. Add, all right, 3 more. Okay, now remember that you are diluting. Okay, they're already giving you 1, so we have this concentration. Find the concentration. So this is 1%. Tenfold, divide 1 by 10. The next should be 0.1. Okay, because it's tenfold, right? The next 0.1 divided by 10. Okay, that's 0.01. But I want you to pause the video and try this thing. Okay, so the next will be what? 0. Pause the video and try it by yourself. 0.001. Should have 5 values. This is 4 now. The last one should be 0. Okay, 0, 0. Okay, 0, 1. Yeah, good. So that is it, guys. Yeah, so if I'm diluting, I'm moving from here all right, from 1 to 0 point, okay, 0.1. So this is my C2. All you need to do is calculate your C1. That's what. And then this is serial dilution. C1 is the same. And of course, all you need to do is calculate your V1. Sorry. Now, this is my V2 10. Okay. So final volume should be 10 for each of them, right? So 10, 10. What I'm looking for is V1. Okay. That's what amount V1 is what I'm looking for. And don't forget the formula. Okay. So find me the V1. So remember that the dilution formula, right? We know C1, okay, V1 equals what? C2, okay, V2. So we're calculating, looking for our V1. So therefore, our V1 will be what? Yeah, V1 will be C2, okay, V2, all right, all over C1. So once you get your first V1, you're good to go. So my V1 here, if I'm moving from 1% to 0 point, okay, so I'm moving from, so this bigger will be 0.1, this is 1, this is 1, see it's 1%, so I want to use it to produce the next one, 0.2. So this is my C1, is my C2, my V2 is 10. Okay, so yes, so now C2 is 10. Do it, you know how we do it, guys. Sorry, C2 is 0.1, yeah, times V2 is 10. Let me clean up. Yeah, so C2 is 0.1 because that's what I'm forming using 1. So 0.1, all right, times 10, okay, which is my V2 all over 1. Okay, so that would be what? 0.1 times 10, that would be, wouldn't that be what? That would be 1, right? It will be 1. So that means 1 cm3. My V1 is 1 cm3. Okay. Yes. Okay. So what am I to write here? Well, what I'm going to write here is that I'm taking, okay, 1 cm3, right, which is what you're supposed to write here. Okay. So what are you taking? I'm taking 1 cm3, all right, of 1%. Yeah, because that's a stock, 1% of S. Okay, so of S, and I'm adding how much of water? Remember that V2, all right, is V1 plus water. V1 plus water. So if I found my V1 plus volume of water, my V1 is 1 cm3. So V water will be 10, which is V2 minus 1, which is 9. So I'll be adding here 9 cm3, okay? 9 cm3 of water will be added to that 1%. 9 cm3 of water, and here water is W. Okay? So this is 10. V2 is 10, you know. We have calculated our V1, right, which is 1. So, and our V water, which we now know to be 9, because it will be 10 minus 1, okay, which is 9. Okay, so it's easy. So the same amount will be taken, same 1 cm cube of that 9 cm cube of water added. Can you see? So once you calculate the first, the next is the same 1 cm cube, right? This is 9 cm cube of water. So you just write it like I've done the first one. All right. See, this is like about 3 marks, guys. Okay. So now, of course, this will be, by the time you remove this is 0.1. By the time you remove 1 from it, 1 out of 10, because 1 plus 9 gives us 10. Remove 1, what will be left is 9. So what will be written at the bottom here will be 9 cm cube. That's what will be left. 9 cm cube of what? Of 0.1%. So that's 0.1% of S. Okay. So what you should write here is that you are taking 1 cm cube of 0.1 here, 9 cm cube of water added. Okay, of course this is going to be 0.01. This will be 9 cm cube of 0.01, 9 cm cube of 0.001. This will be 10 cm cube of 0.001. So you are taking the same amount. Well, if you are moving from here to here, since this is 0.1, so that will be 1 cm cube of 0.1% plus 9 cm cube of water. Okay, 9 cm cube of 0.0001. Okay, 0.001. All right. So look at it. Label under correct sequence speakers 1 0.1. So just by labeling this, you got 1 mark. Show transfer 1 cm cube to each under 1 mark. Show 9 cm of water added. So 3 marks added. 3 marks. Then the next question goes. These are a sequence of instructions. Carry out 1 to step 6. Step 1 to step 6. They say prepare the concentrations of star solution as shown. They said they use a glass rod to mix the star solution and water. We've done the label test tubes with concentrations of star solution. Put 5 cm cube of each concentration of starch solution into an appropriate label test tube. You just follow the instruction, guys. Put 3 drops of iodine into each of the test tubes. Shake gently to mix. After step 4, observe the color of the liquid in each test tube. You may see the same color in more than one test tube. It may help to hold a piece of white card behind the test tube to see the color clearly. Yeah. So that's what we're going to see, right? So fig 1.3 shows the key you need to use to record. So this is called if you see blue, black, put how many symbols, how many pluses? 1, 2, 3, 4, 5. So blue, 4 pluses, dark brown, 3 pluses, brown, 2 pluses, yellow. So you see that the higher the concentration of starch, right, the more crosses, the more of the symbols, right? This has the highest concentration, this has the lowest concentration. Record the result in AII using symbols shown in the key. All right, so if you're going to record it, guys, how do you record it? You carry out the experiment, but if you're going to record, use the right table. Remember, in recording, always draw enclosed table. Okay, so this is the heading. Okay, the first column is for independent, which is concentration. This is concentration of S. Okay, so concentration of S, right? Concentration is the independent variable of S. Okay, now you write the unit. Do not write the unit inside the table, write it on the heading. Okay, this is symbol. Symbol is what you're recording. There's no unit for symbol, guys. So let's go. So you see, based on what you observe, we have about 5 values or thereabouts, okay, for concentration. record the symbols. So you have to draw cells like this. All of these are very important if you're going to have your mark. So this is 1, this is 0.1, okay, that is 0.0.01, 0.001, 0 point. These are different concentrations you are going to test, right, for the color change using iodine solution. You write the symbols there. Remember, the trend must flow. It must follow the right trend. That means the highest concentration should have the highest pluses, right? Like 1 should have the highest pluses compared to lowest. So you write if this is plus, depending, okay? You must follow the trend. So record your table in appropriate, okay, table. That's, it must be enclosed. So you see it. Form 5 marks. Heading for independent variable percentage concentration start before. So this heading here, 1 mark. Yeah, dependent variable, no units, okay, 1 mark. Heading for dependent variable symbol, see 1 mark. Symbols for all samples, and that mark. Results for the highest concentration of starch, more than for the lowest. See, more symbols than highest and lowest, okay. Result recorded by the highest concentration of starch, more than, okay, the next concentration. So guys, you must follow. Once you follow it, right, this is, you're going to do the practicals. So based on the color change, but the trend is that the higher the concentration, the more of the symbols, okay. All right, so let's go see the key again. Show the key you need to record your result. Record your results for X and Y using the symbol. So you have to record it, right? So record it based on your table, what you have done, record, okay, for X and Y, just 1 mark. A student investigated the hydrolysis of starch by amylase. This is testing your ability to analyze and evaluate. The student decided to use a semi-quantitative method to measure the product. This student decided to use semi-quantitative method to measure the product. These products are reducing sugars. So just how the student would use a semi-quantitative method to measure the concentration of reducing sugars. Okay, this is reducing sugar. If you are using a semi-quantitative, you can use a curve. Okay, so you write then. Anyway, pause the video and try it. Okay, but make sure you are writing that you're going to prepare solutions with known concentrations. Okay, and then when you prepare solution with known concentrations, you carry out Benedict tests, okay, and then measure time for color change, and they use it to plot graph. Then you can now carry out the same Benedict solution on the unknown concentration, okay, and measure the time for color change. Then go to the graph you have plotted the standard curve, and then use the time to extrapolate the concentration. Okay, yeah. So first of all, use known concentrations. Plot a graph. Remember the concentration should be, of course, if you use known concentration, you measure time for color change. Concentration against time for known concentration. So this graph, as concentration increases, time for color change will decrease. So it's going to have maybe something like this, a negative curve. So your known concentration U, okay, carry out the same Benedict test, and then dictate the time. All right, when you get to the time, extrapolate, trace the time for color change to the curve, and when you get there, take it down. Then that will give you the concentration. So this is your concentration of the unknown sample. Okay, so you see it. Benedict solution, Benedict reagent, time for color change, or prepare known chart, 5 reducing sugar concentrations. Compare unknown time for color with known concentration time. You can compare it, color change, or the known concentration. The ones that have the same color time for color change should have similar, or use this curve. Okay. All right. 3 marks. One resin sugar is produced by hydrolysis of starch in maltose. Maltose is a disaccharide that can be hydrolyzed to glucose by the enzyme maltase. A student investigated the effect of maltose concentration on initial rate of reaction. All right. The results shown. This is the result here. Plot a graph, okay, of the data on the grid. Use a sharp pencil. I want you to plot this graph. Okay. Remember that if you're plotting a graph, the x-axis should have your independent variable, which is concentration. Okay. So write concentration here. Write the unit, mole per dm cube. Your independent variable initial rate of reaction should on the y-axis. Write the unit. Okay. Now in biology, we like to use 2 cm. Okay. So choose a scale for this. If you choose a scale, 2 cm is 10 boxes, 10 small boxes for 1, right, mole per dm cube. These are 2, 3, 4. All right, and then this. So if I'm going to plot this graph, for example, of course, use a ruler. All right, do I have to rule this? I don't have to. Okay, but you have to rule it. Okay, so you rule it and then you label. Okay, label well. So this is going to be concentration. Remember to write the label and put the unit. Okay, yeah, mole per dm cube, mole per dm cube. And this is rate of reaction. Make sure you write, okay, what is there, right? What is here on this. Now on this, I choose my scale. This is 1. Okay, this is 2. This is 3. Okay, and that's 4 and 4. So this content was the highest is the highest value there is 3.95. So 4 covers it. Okay. Now here the highest value here is 260. If I try to do 54, remember we use 2 units, 10 boxes, 2, okay, 50. This is 100. Okay, so I'm plotting this graph with you. Okay, the next one is what? 150. 150, right? Yeah, and of course this is 200, 200, and that is what? 250. Okay, and the last one there is 300. That's 300 is somewhere. Okay, so remember that one box here is 0.1. One box here is 5. Okay, so if I'm plotting 0 and 0, so 0, 0, that's going to mark, okay, point here. The next one, 0.6, will be count 6 boxes. Okay, for these 6 boxes, 1, 2, 3, 4, 5, 6 should be here, right? Against what? Against 90. So take it up. 90 should be it should be a box here. Yeah, somewhere here. 0.6 and 90. Check here. Shouldn't it be here? Circle it. Okay, is this correct? Yep, 90 should be okay. Each box is 5. This is 6. 0.6 should be here. Okay, I think there's a little mistake there, so let's bring it. Yeah, see, so it should be somewhere here. So just circle it. So next one is 1.3, 175. 1.3 175, 1.3 175, 175 should be in the middle. Okay, 1.3 should be here. Okay, what do you think? Okay, the next one, 1.85 225, 1.85, 1.85, what? 1.3, okay, 1.85, one values. Okay, and this is 1.2, 1.2, okay, 1.85 and 175. Nope, 225. 225 should be in the middle here, right? Yeah, 1 2 3 4 5. Okay, 1.85 somewhere here. Okay, yeah, somewhere here. Okay, I know that I'm not teaching you how to plot graph because we're supposed to have learned it. Now 2.45 in the middle between 2.4 and 2.5 and 255. Okay, now this is 2.45. Yo, pum, pum, pum. 1 2 3 4 255 somewhere here. So check. Check here. Okay, no, it should be here. Yeah. Okay. And then 3.55 260. 3.55. Yeah, that's 3.55 260. That's the next row. 3.55, 3.55 somewhere here. Okay, 260. Okay, then we have 3.95 260 again. 95 somewhere here. Okay, so you can see that the graph is beginning to, yeah. So that means 260 is our initial rate of reaction. Sorry, is our Vmax. Okay, is our Vmax. If you trace this, yeah, oh boy, trace this 260. So this is our Vmax is, so Vmax is, okay, 260, which is 2 boxes. Yeah, this is our Vmax. Vmax is 260 Vmax. Okay, but before then you have to connect the dots. Connect this and this. Connect. You can see that it's increasing as, right? Concentration is increasing, rate of reaction increase. Connect the dots. Connect the plotted. Okay, the plot. Yeah. So that's our Vmax. Okay, our Vmax 260. Yeah, 260. Yes, yeah. Okay. So 260 divided by, because you know that Km, in case you're asked to find Km, how do you calculate Km? Km is half Vmax, right? So that is half substrate concentration at half Vmax. Half Vmax is half of the sub of enzyme active site occupied. So half of Vmax is half of 260. If 260 is a Vmax, that's maximum rate of reaction, divided by 2, that will be 120. Is that 120? 130. So 130, you go to the graph and extrapolate. So 130 should be somewhere, this is each one, Okay, 130. What should be 130 here? This is 100. Each box, of course, the middle is 125. Here, 130 is a box above 125. Okay, so somewhere here, 125, 130, 135. Okay, yeah. So this is half Vmax. Okay, yeah, half Vmax. Yeah, this is what you might be asked to do in exam. Half Vmax. Okay, then if you want to find your Km, trace it to the curve. Yes, let's trace it to the curve. Can you see where it touches the curve? You trace it down to, so this is 1. This one is our Km. So Km value is this one. This is a Km value. 1 is a Km value. That's also concentration, 1 arbitrary unit. Okay, 1 arbitrary unit. I hope you understood how we did it. 1. So I plotted the graph, don't get the Vmax Km value. Okay, so let's see what the math scheme says. The math scheme says answer. So look at it. What should be in your, okay, look at it. X-axis concentration of maltose should be here, and y-axis initial rate of reaction should be here. That's 1 mark. Scale for x-axis, I told you 1 mole 2 cm must be used. Okay, 1 mole is 2 cm, and for the other one, 5 arbitrary units to 2 cm. Yeah, 50, sorry. So that's another mark. Then correct plotting, 1 mark, and of course join the plot, another mark. Can you see? We've done a fantastic job because you are asked to do this in exam. Okay, all right, now 4 marks, guys. Use the graph to estimate the Michaelis-Menten Km. We have done that. Show your working. We have done that. That's 1 more. Okay, our Km value, we've shown it, which is 1. Go back to the graph. Okay, yes, correct. Vmax, you see how we did it? Calculation of Vmax, I mean 260 divided by 2, that's 130. We checked it. Km was 1. Okay, so 2 marks for that. A student investigated a different enzyme, all right, and found the Km value. Now the Km value of this enzyme is 0.5 mole per meter cube. State which enzyme, Z or maltase, has a lower affinity for its substrate. Give a reason. Guys, remember that our substrate, okay, this is 1. Okay, yeah, 1. But remember that Km value, okay, remember this principle that states that Km value is indirectly proportional to enzyme affinity. So that means the higher the Km value, the less the enzyme affinity. So enzyme affinity. So enzyme affinity, I call it enzyme AFF. So enzyme affinity is the tendency of the enzyme to bind to its substrate. Okay, so the one that has the lower Km value will have higher affinity based on this relationship. So and Z has 0.5. 0.5 is lower than 1. So Z has, all right, should have no, the one that has low affinity is the one that has high. Of course, yes. So the one that will have low affinity is our maltase enzyme because it has higher Km value. Okay, the Km value is 1. 1 is higher than 0.5, so it will have lower affinity for its substrate. Did you get? Yeah. So maltase has, because it has higher Km value, 1 mark. Using graph, explain the change in the rate of reaction between. Now, of course, you know that as positive and try it. As substrate concentration increases, the rate of reaction will increase. Okay, why? Because more substrate available, so more effective collision between the enzyme's active site and substrate. Now, but at low substrate concentration, you know, there are fewer substrates available, so low rate of reaction. Now, but at lower substrate concentration is the limiting factor. Okay, but you know that the graph will reach um, it will reach when it reaches levels of, that means active sites are saturated. That's at Vmax. So you see it, more saturated molecules, more circulation, more enzyme complex. When levels of, that means active sites are saturated. So enzyme concentration is now the limiting factor. Okay, thank you so much for hanging on. Right? So we'll see you in checker 2. That's 2024 May June 31 series. Okay, see you. Thank you for the ride. Wish you the best in your exam. Bye.